Here's the conclusion to identifying carbohydrates lab. If you remember, we had a series of 8 test tubes, and we were testing for monosaccharides and polysaccharides. We were testing for monosaccharides with this blue stain. This blue stain is called Benedict's solution. We also were testing with this golden or orange stain called iodine, and that reacts with these polysaccharides. So we're going to go through the lab, and we're going to indicate everything that we had from the beginning, and then we're going to see all the color changes that we had as well. So we're going to go ahead and list the molecules present if you haven't already done this. So if you remember in test tube number 1, we had glucose. Glucose is a monosaccharide. Test number 2 has sucrose, which is table sugar. That's a disaccharide. And then number 3 has starch, and starch is a polysaccharide. And then number 4 had a combination of both starch and also some spit. We then put a blue stain. This is the Benedict stain. Okay, so initial color of the stain was blue. So you can put blue, 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 and blue. We'll get to that stain in a little bit. Second experiment with the iodine. Test number 5, we had water. Number 6 had starch. 7 had starch and spit. And 8 had glucose. So it's a very similar experiment with this compared to this. But again, here we're testing with the blue Benedicts to test for the monosaccharides. And here, the second one, we're testing with the iodine to test for the polysaccharides. So here's the analysis down below here. Benedict solution, when heated, binds to monosaccharides. So if you remember, here's a monosaccharide, and here's the Benedicts. It binds to the monosaccharide, and so that when it is heated, it changes the stain from a blue to a range of colors that include green, yellow, orange, red, brown, based on the concentration. We didn't have enough time during the duration of the experiment to show all the color changes, but we did see a color change, and we'll examine that in a few minutes. While it does bind to monosaccharides, it does not bind to polysaccharides, meaning it does not bind to this molecule. Okay? In the chart above, indicate monosaccharide present if the color after the stain has changed to indicate no monosaccharide. Okay, so this is what we're going to be doing. We had the final color of these. So let me show you this one right here. This was sucrose, again, and sucrose started off like this, it ended like this. However, the glucose started off blue, and it ended up looking white. So the final color of this was white, and this stayed blue. So if there's a color change, that means that we have this scenario present. We have the blue stain reacting with a monosaccharide, which occurs with glucose. That's the color change. If there is no color change, that means there's no monosaccharide there. So what you can put over here is that we can just abbreviate and say we've got, we have monosaccharide. We can write the whole word just to get some practice. It's a monosaccharide. So we had no color change, which indicates that there's no monosaccharide. So we're going to abbreviate for this one. We're going to say there's no mono. Why is there no mono? Well, because if you take a look at here in this chart, we have different types of carbohydrates. A monosaccharide such as glucose. We also have a disaccharide, which is a dimer, and that's going to be sucrose. So our sucrose is a dimer. It is not a monosaccharide. So there's no monosaccharide that is present, and that's because sucrose is a disaccharide. So we have disaccharide present, but we don't have a test for that. The third test tube looks very, very similar to the first. This one was filled with starch. And notice that there's no coloration, no difference compared to this one right here. Maybe a slight coloration difference. But the real difference is when we get to the last test tube right here, and we see that the one with the spit definitely changes color. So the third one stayed blue, okay, and this is going to indicate that there is a negative reaction. So we have our blue stain and we've got our starch, okay? It doesn't react with that, so it means it's going to stay blue. So there's no monosaccharide for this one either. And the reason why there's no monosaccharide is because we have a polysaccharide. And the last one changed to a white color. And again, the reason why is we had starch. So 4 should take a look, or should look very closely to number 3, but again, we added the spit. And the spit act as the scissors to chop up the polysaccharide into monosaccharides. So of course, we're going to be having monosaccharides present. So we're going to be saying that, yes, we have monosaccharides present, and you can go ahead and abbreviate that one. Those are the results for the first experiment. So in the second experiment, if you remember, we started off with something that looked like this. This is an orangish color. So I'm just going to say, it's kind of orange, red, gold, brown. I'm just going to say gold. So iodine starts off with this golden color. And again, we're looking for a difference in color change. And so this is the water, okay? And the iodine is testing for a polysaccharide. So here's the orange stain. We're looking for these chains of glucoses like this. And it's going to be binding onto a polysaccharide such as starch. But there's no starch in here. So this stays gold. And this indicates that this is a negative reaction, meaning there is no polysaccharide. So I'm going to say there's simply no polysaccharide present. Taking a look at the next one, we have our starch. Okay, you can actually see there's a slight bluish, purplish coloration to that. Very different from this. This is showing we have a positive reaction for starch. This one turned purple. Why did it turn purple? It's because we have the starch as a polysaccharide. So there are those two. And then number 7. Here's number 6 with the starch. Here's number 7 with the starch, but this is the one that we also added the spit to. You can see that there might be a very little bit of, tiny bit of starch left, but most of it's gone, and we're left with monosaccharides in there. So this stayed gold. Okay, and the reason why? Well, it started off as a polysaccharide, but we're going to say the polysaccharide was digested. That's what your enzymes do. They digest things into smaller things. And the last one, this is the glucose. Looks very similar to the water, and so it stayed gold. That shows a negative reaction, and the reason why is because it's not a poly. We don't have polysaccharides. There's no polysaccharide present. The reason why is because glucose is a monosaccharide. So I'd like you to now turn this over to the next page. And we're going to answer these questions now. Based on the coloration, explain why there's a difference between test tube number 1 and test tube number 2. Okay, test tube number 1 has glucose, number 2 has sucrose. The blue stain binds onto monosaccharides. So in test tube number 1 is glucose. Glucose. Okay, and glucose is a monosaccharide. So what happens is, because it's a monosaccharide, it reacts with the blue stain. And we're just going to call it the blue stain rather than Benedict's. Number 2 is sucrose. What type of sugar carbohydrate is sucrose? Sucrose is a disaccharide. So since the blue stain only reacts with monosaccharides, here's a disaccharide, it's not going to react with the disaccharide. We have this, it doesn't work. So it doesn't react with the blue stain. Doesn't react with the blue stain. Explain why test tube number 3 does not react like test tube number 1. Let's go back to our chart. Test tube number 3 is starch. Well, that was a negative reaction. It went from blue to blue. The reason why is because there's no monosaccharide. So we can say number 3 is starch. And again, starch is a polysaccharide. And this doesn't react with the blue stain. Number 3 is starch. It's a polysaccharide. Okay? Does not react with the blue stain. Explain what's causing the color in test number 4 compared to number 3. So if you remember number 4, this is the starch that had the spit in it. And we did see a color change. Okay, number 4 looked like number 1 with the glucose in it. So that means we must have been producing some monosaccharide in this. So number 4, it has starch plus saliva. And the saliva has enzymes. So it has starch plus saliva with enzymes. And again, these enzymes act as scissors that digest, that cut the polysaccharide, which is the starch, into monosaccharides of glucose. And it's these that are reacting with the stain. That react with the stain. So again, number 4 had starch and saliva with the enzymes. The enzymes chopped up this giant molecule into the monomers of glucose, and this reacts with the stain. That's the first experiment. Second experiment. Explain why test tubes number 5 and number 8 do not turn this purplish color. Why do they not look like this? Well, what's in test tube number 5? Take a look. Look at your chart. What's in test tube number 5? Number 5 is water. And number 8 is glucose. Neither is a polysaccharide that will react with iodine to react with the iodine. This is the orange stain. Okay, so we've got glucose, doesn't work. Water does not react. Explain what happened to test tube number 7 to create a difference in color compared to test tube number 6. So number 7 had the starch, but also had the saliva. So we would expect, since it has starch, to stain purple. But again, the saliva chopped it up. So this is a very similar answer to this. This has starch, oops, up here, has starch plus saliva with enzymes that cut the poly into mono. Cut polysaccharides into the monosaccharides. So after time, what happens is all of this giant polysaccharide gets chopped up, and there is little or no polysaccharide left. So this has starch and saliva with enzymes that cut the polysaccharide into monosaccharides. So after time, there is no polysaccharide left to stain. And the last of these questions, based on the color, type of molecule, what type of molecule is glucose? Glucose is what type of sugar? It is a monosaccharide. What type of molecule is starch? It is a polysaccharide. So hopefully this enabled you to become more familiar with monosaccharides, disaccharides, and polysaccharides. All three of them are carbohydrates. One is a monomer, one is a dimer, and one is a polymer.